I'm the next person from the Wroclaw University of Environmental and Plant Sciences, and today I would like to speak about the hydroxylation reaction, but mainly focus on the biocatalyst which I used for that reason. But first, let's focus on the compound. So that's the flavonoids. And analysis of the structure-activity relationship revealed that the presence of the catechol moiety significantly increased the activity antioxidant capacity, which is closely related with that flavonoid health promoting activity. So due to that, the main goal for my research work was to create the compound with the C7, C8 catechol moiety and C6, C7 catechol moiety. So I would like to divide my presentation for these two compounds. So let's focus first on C7, C8. And for that one, I follow that's 5 main steps. The first two was about the sequence identification and also the overview of those amino acid structures. And the next one, that's the purely genetic part of my work. And to last, that's about the enzyme characterization, reaction condition assay, and also product identification. So 3 enzymes, flavin-dependent monooxygenases, was selected for that task. The first one from Rhodothrula glutinase, based on the next generation sequencing, and next two based on the available database and the literature. The first one from Herbaspirium serapedica, and the second one from the Pseudomonas putida. So let's start with this one from the Rhodothrula glutinase. And as I said, we run the NGS for that enzyme using Rhodothrula glutinase culture induced by naringenin, and as a control, the culture without any inducer addition. And after that, based on the differences in the expression, and also based to the comparison to already described C8 hydroxylase, we was able to select 5 potential sequence for our further investigation. Moving to the next enzyme, that's the one which was already described in the literature, but in this presented work, researchers were mainly focused on naringenin degradation pathway, and they did not extend the research about that particular enzyme, so we decided to do that. And the last one, that's from Pseudomonas putida, for which we run the comparison, including the sequence from Rhodothrula glutinase and Herbaspirium serapedica. And after that, we find one common match, which we also choose for our next test. So after this all work, including all sequence selection, we decided to clone each of them together with the J promoter and run in vitro assay using naringenin as a substrate, and after 24 hours and extraction, we verified the results based on the UPLC analysis and compare our results to the commercial 8-hydroxynaringenin standard. And that one revealed that FD was active against naringenin, and we also was able to identify 2 new C8 hydroxylase of naringenin. So we know that, so we just took step back and look one more time to the amino acid sequence, and that revealed that All of them contain some characteristic motif for that FMO enzyme group, the Rossmann fault, and also GD1s. So next part, that was about the similarity between them, and that's all enzyme was classified along another's enzyme also catalyzed CA hydroxylase of flavonoids. And especially this two bacterial one was really was classified like really similar to each other. So then we moved to the induced expression system, in which we based on the Ramnow's promoter, and also we used this N-histag for the purification assay. And after that, we was able to verify that all of these three enzymes we are able to produce in bacterial cells. So that was quite nice for us. And also each of them required another pH for their activity. So that was the first biochemical analysis, and then we verified their reaction condition for each of them, and they look quite similar. But what was surprised for us, that's the volume of pH, because all of them was preferred the 5.5 pH. So we also detect that three of them was not quite stable in that pH. And according to the stability test, we decided for the next part used to pH at 7.5. And until that, the all enzymes look quite similar. So the first differences between them. So that's about the coenzyme addition. Only for the enzyme from Rhodotorula glutenis, we achieved that the extra addition of the FAD increased the naringenin conformation. For the enzyme from Pseudomonas botida and Herbaspirillum serrapatica, we did not observe any of that results. So even more noticed difference between those enzymes. That's the yeast one, except only flavanols and flavones as a substrate. And these two bacterial ones extend the panel of the substrate with flavanols and isoflavones. So that was the first, that was never before described in the literature. So we, of course, we wanted to upscale that reaction. And then we verified the problem with the stability of the product and also of the substrate. Because even if we used only the bacterial cells without any plasmid, some of the substrate were degraded after 24 hours, especially quercetin. Yeah. So that was visible even more for the product of the hydroxylation, because after 24 hours, not even once was detected. And that for the quercetin is especially visible because I was able only detect some product after 6 hours, but it was not satisfied at all. So we decided to switch to the in vitro reaction and manipulate with the redox potential, for which we chose 4 compound, and that gave us results that the dithiotrietol was able to increase the stability, improve the stability of our product. So we ran with the test using 3 different substrate and without DTT addition. After the 1 hours, we was not able to detect the product, but with DTT, the product was still visible. So as a proof of the concept, we decided to run 24 hours reaction using quercetin. And for this one, we achieved that using FDEE, was able to produce 0.16 and with PPF8H 0.67 gram per liter of the gozifetin. So that's confirmed that approach worked quite nice. And now I would like to switch to the second catechol position, that's C6, C7, for which one we decided to use different group of enzymes. So that's the P450s monooxygenases. We choose both already described in the literature, active against naringenin, and also this first one from Scutellaria biocatalysis was already used in development of bicalane synthetic pathway, but with the conversion of quercetin around 1.7%, and also required reductase for that group of enzyme activity. And bearing in mind that there's the problem with the eukaryotic protein expression in E. coli, we also change the workflow. So after the amino acid sequence selection, we decided to use 3 different expression strategy. The first 2 based on the plasmid expression, in which in the first separate gene were expressed, so separate protein, of course, and the second one, the fusion protein was involved, and the next part, which I involved to use the SIGA platform to incorporate those genes into bacterial genome. So today I would like to talk about these 2 first approaches. And because of these many challenges with this eukaryotic protein expression in bacteria, and also after the in silico analysis, which revealed that both of that enzyme contains an transmembrane fragment, we decided to start with this N-terminal modification library, including translation and sequence alteration. And that graph clearly showed that without any N-transmembrane fragment, an enzyme from Scutellaria biocatalysis achieved the best results. And addition or exchange of the transmembrane fragment was also helped, but not as much as we expected. And especially when we just look at the second enzyme, for which the exchange of that fragment results inhibited of their activity. So for the next test, we decided to use only these 2 with the nice bicalane production. So the next part was about the E. coli strain selection, and that was also quite surprised for us, because that revealed that the H5 alpha was the best host for that kind of the enzyme, even if we find in the literature that especially the Rosetta strain was dedicated for that enzyme. So knowing that, we focused about the media selection, and for that test, a modified version of TB named dynamic media gave us the best bicalane production, and the last improvement, that's the media supplementation, and that's well-known addition of acid for that enzymes group, and also that revealed that that can improve the amount of the product formation. And one more time, we verified the two types of the strains, and still DHI5 alpha gave us quite nice product concentration. Okay, so that was about the first approach for that kind of the enzymes. And based of all of that improvement, we was able to achieve the conversion of the quercetin around 15%. So that's, in comparison to the original paper, quite nice results. So let's move to the second approach. That's about the fusion protein. That's when we wanted to express the hydroxylase and reductase joined by the linkers, and that linkers were selected based on the literature and of course based on the website dedicated for to design for that genomic parts. And we select them based on the type, but or based also on some literature where they was used for exactly the same group of the enzymes. And then we clone that together with the reductase, and also for these two terminal variants, which we gave the best results from the first approach. And after the transformation, we was able to achieve around 100 clones for both of the variants, and from among 40 was tested in vivo assay. And for the first variant, 90% of the clones were active against the chrysin, but for the second one, not even once. So now let's just focus about these positive results, during which the conversion was around 0.5 to 13% of the chrysin conversion. So for the 9 clones with the best substrate conversion, we also decided to run with the SDS page analysis, and expected size for the protein, that's supposed to be around 130 kilodaltons, but as you can see, we're still able to detect the separate protein. So that's also which we need to verify at that moment. And also the first thing which we need to check, that's the linker type, which we already have for that clones, because some of the linkers can also be responsible for that enzyme separation. But according to that test, we was able to achieve the conversion around 30%, still better than the original papers. And to conclude this presentation, I just wanted to say that different expression analysis as well as analysis of the sequence homology allows the identification of the FMO sequence. The yeast FMOs accept flavonoids and flavones, while the bacterial one extend that panel, including isoflavones and flavonols. The manipulation with the redox potential could stabilize the product formation after the hydroxylation. P450 enzyme can be expressed in E. coli bacterial cells in active form, and also that the N-terminal modification and the addition of 5-aminolevolic acid improved their activities. And in the very end, I would like to thank my supervisors and my colleague for their support, and also the community for that, to accept my speech for today, and your great audience for your kind attention. Thank you very much.